So as Sheetal mentioned, my job today is to put into context why this prize was given to this particular discovery. And in order to do that, I will give you a little bit of a background into the study of circadian clocks. So clocks must perform two important functions. Okay. So clocks must perform two important functions. One is that they must measure, can you hear me? Yeah, that they must be able to measure the passage of time. And secondly, they must be able to give local time. These are two things that any clock must do. But today, we are going to talk about clocks that measure a duration of about 24 hours, and that's why they are called circadian clocks. They are basically biological entities that drive rhythms that are approximately 24 hours. So the word circadian comes from the two words circa and dian, or dies a day, and sorry, dm or dies, both of which mean a day, and they refer to, as I said, mechanisms that exist inside biological entities or inside organisms, sorry. Often we come across people using the word circadian rhythms and circadian clocks simply to refer to processes or behaviors that show day-night variation. But I want to tell you that that's not always, that doesn't always mean that they are controlled by circadian clocks. In order to say that some process or behavior are controlled by circadian clocks and that that process is indeed a circadian rhythm, one must look at a few characteristic features. Those I will describe just now. One important thing is that rhythm that is driven by an internal circadian clock must persist with this near 24-hour periodicity, even in the absence of external time cues. Because otherwise, you could have a 24-hour rhythm or a day-night rhythm simply because that rhythm is due to environmental cycles, because light, temperature, and everything else around us changes simply because the earth rotates around its own axis. And therefore, organisms might simply mimic what is happening outside, realize that it's light outside, and do a certain behavior during the day or night. However, in order to know whether that behavior or process is controlled by an internal clock, one must also demonstrate that that rhythm persists even in the absence of external cycles. And it is attributed to this French astronomer, he was not even a biologist, that the first demonstration that something like this, some kind of internal entity might exist, the demonstration is attributed to this person, who observed that these Mimosa pudica leaves, the touch-me-not leaves, which showed a day-night pattern in their opening and closing, in that at night their leaves are closed, seems to persist when he takes these leaves down into a chamber where there was no information about light. So they were kept in a basement where he proposes that no environmental time cue is reaching them, and yet these plants were able to open and close their leaves as if they knew what time it was outside. And in fact, this astronomer only proposed that these plants possess a mechanism by which they knew what time of day it is outside, without actually talking about there being clocks inside. Now from plants we move to a rhythm that's observed in humans. Between plants and humans, almost every other organism that people have cared to look at seem to possess circadian clocks. Here is a rhythm of plasma cortisol that has been observed in humans, and this is a rhythm that is observed in humans that are kept in a chamber, sorry, not a chamber, in a lab condition where every other environmental factor is kept constant except light-dark cycles. So the yellow shading depicts the fact that these humans are experiencing light, while the gray shading means that they don't have light information. So this is noon depicted by N, and midnight depicted by M. So there are 3 days in the life of these humans, and plasma cortisol is being plotted here on the y-axis. And you can see a nice up and down pattern in plasma cortisol levels. And if you allow these people to now go into a condition where there is no more control over day and night, in that at least they don't have information about day and night, and so these humans are now in a chamber where Everything else is kept constant, temperature, humidity, food, they are not even allowed to move around a lot. They are going to be given the exact same meal every 1.5 hours or something. So it's a pretty boring situation. However, you can still see that their internal body clock seems to be able to keep time. And now that the periodicity in the plasma cortisol is approximately 24 hours. The fact that this rhythm has persisted even in the absence of external time cues suggests that there must be an internal mechanism, and so that clock must be of endogenous origin. Now, it's not enough for you to have a clock with a near 24-hour period. That clock must be able to entrain or synchronize to external conditions. Only then it's a useful clock. So what are these environmental cycles that can entrain internal clocks? It turns out light is a very important time cue for clocks. Also, there are other cues such as temperature, humidity, possibly even the presence of food, predators and mates, depending upon the organism and the environment in which it lives. However, again, often there's the misconception that anything that cycles outside in the environment can potentially act as a time cue, not necessarily. In order to know whether that time cue actually affects or whether that environmental cycle actually impacts the clock, one needs to do more than just observe that this is cycling, that a behavior or a process is cycling along with the environmental factor. One must ask whether there is truly entrainment of the internal clock, and circadian biologists do that by looking at whether that relationship that rhythm exhibits in relationship with the environmental cycle is stable, whether there is some anticipation of how environmental cycle changes, and so on. We can discuss more about how you can rule out whether this is merely a masking phenomenon or truly entrainment of the internal clock to the environmental cycle. So, but this is an important characteristic of an internal clock. Third important feature for a circadian clock is that its period, the speed at which it runs, must be temperature compensated. What do I mean by that? You can imagine that if you are talking about a clock which is made up of biological molecules, which will be composed of biological reactions, they could be susceptible to changes with temperature, because almost every biological reaction that we know is its rate is changed by temperature. But that would not be a very good system if you are going to have a clock whose speed changes with changes in the environmental temperature. And in contrast to most biological reactions, it is found that indeed biological clocks are the period of biological clocks are temperature compensated, in that even up to 10-degree differences in temperatures produce very little differences in the speed with which clocks run. This graph simply shows you the contrast between how metabolic rate changes for several reactions with temperature as compared to biological clock period, which is very fairly temperature compensated and quite stable across a wide range of temperatures. You can extend that changes in temperature. You can think about it as changes in energy levels, and people have shown also for certain kinds of clocks that clocks are also nutrition compensated, in that levels of nutrition don't affect the speed with which clocks run. So this is the third important property of a circadian clock. One other property which I will simply state as of now is that usually the speed in the clock or several properties of these clocks are reproduced across different levels of organization, at the level of an organism, at a sub-organismal level such as in a tissue, at the level of the cell, and even at the level of molecules. For now, just take this as a statement of fact, and hopefully it will come up as we go along over the next few minutes. So here, so far, I was talking about how one might look at analogies between how a physical clock works and how might we compare that with internal biological clocks. But for the longest time, people who were studying clocks were looking at them at that level. Then questions began to be asked about what is it that these internal clocks are made up of. It used to be thought of in this form, where there is a core clock that we don't really understand, but we know that there are environmental inputs to the clock which drive changes within this internal clock and gives rise to outputs in the form of rhythmic behaviors of its logical processes. But it was several people were interested in what this core molecular, what the core clock itself is made up of, and therefore what might be the molecular and cellular basis of these clocks. But before people actually did experiments to ask that question, it was predicted that these clocks might be generated by feedback loops. And if I had given you 5 minutes, you might have come up with that as well, knowing that in order for you to have self-sustained oscillations, it's probably very useful to have feedback loops. So where there would be the action of certain processes having an effect which in turn feeds back and also impacts the action in such a way that such a loop could be sustained for a long time. So these kind of feedback loops could occur at the level of transcription within cells, at the level of translation, maybe post-translational modifications, and even feedback from the cell membrane to the internal components of the cell. So these ideas were around. However, there was no hard empirical evidence to show that indeed such mechanisms may be what has generated the circadian clock. So the first clues for looking at molecular and genetic underpinnings of circadian clocks came from the work of two people, mainly because of the work of Sheeba Vasu, who is also credited with several other findings. And yes, Sheeba Vasu started off as a physicist and went on to look at phage genetics. Some of you may be familiar with him telling us about recombination occurring within genes and fine mapping of genes. And apparently at some point when he had two little daughters, he began to wonder about how is it that these two siblings, full sibs, are so different in their personalities. So you can see what children can do to their parents. So he began to wonder about how genetics shapes personalities, and at that time he thought that it might be possible to use the kind of methods that was then available, which Professor Ranganath also talked about, which is to do random mutagenesis using chemicals such that you can create single or point mutations across the genome somewhere, look for differences in phenotype from normal, and work backwards and ask which genes may have, or mutations in which genes may have resulted in such a phenomena. But people who studied clocks at that time were very skeptical of this approach to look at genes that underlie clocks, and thought that clocks are far too complicated and complex to be able to be dissected out with simple point mutations. But he persisted nevertheless, and due to the efforts of Ronald Konopka and himself, they published this first paper which described a mutation at a locus which seems to affect a rhythm phenotype. So in order to go there, as I said, we will have to look at flies. You've seen this life cycle before. Many processes in the fly are rhythmic, show a daily rhythm, predominantly eclosion or emergence, and the activity rest or sleep rhythms are quite easy to study. So first I'll tell you very quickly about how we study activity rest rhythms before I go back to Konopka and Vasu and then to the Nobel Prize winners. Very short detour. So this is a fly in a glass tube, and you can place such glass tubes into what are known as Drosophila activity monitors. Here is a fly sitting in one such glass tube, and a cartoon will show you how this activity monitor records activity. So in a glass tube with food at one end and a plug at the other, if you put in a fly, there's a pair of infrared emitters and sensors, such that whenever the fly moves across that tube, it cuts this infrared beam, and that is taken as an activity count that's recorded by the computer. And you can imagine you can do that with several hundreds of flies at a time, depending. So each monitor can hold about 32 flies. So you can record the activity rest patterns of flies across several days. And here is an example of this activity rest graph or an actogram. We circadian biologists are very fond of such graphs, and so you will see that in my slide later also. So I'll explain what it means. Here on the y-axis are days, day 1, day 2, day 3, and so on. About 10 days are plotted, and on the x-axis is time of day. So this is 24 hours. So basically you're looking at the life of a fly over these 10 days, and this particular fly has been placed in an environmental chamber where there is 12 hours lights being on and 12 hours of darkness. And you might notice that these flies seems to show two bouts of activity, and they are very predictable across days. Now you can stop providing a time cue in the form of light, and now submit these flies to constant darkness. So over the next 15 days or so, the flies do not have any information about time from the outside. Nevertheless, hopefully you can see that there seems to be a pattern in how the flies exhibit activity and when they take rest. And this will probably be a bit clearer if I take a Xerox copy of this actogram and paste it next to each other, simply to show that over the first few days you see a nice rhythm that's almost exactly 24 hours, and that was because the flies had information about time from the outside in the form of a light dark cycle. And over the next 15 days, when they did not have information about time from outside, they continued to exhibit a rhythm. I hope you can see a rhythm in the fact that there is a periodicity in when the flies stop activity each day, and a periodicity in when the flies start to become active each day. And this fly seems to have an activity rest rhythm and a free running circadian clock with a periodicity slightly longer than 24 hours, because each day that behavior seems to occur at a later time than the previous one. Okay, so this is one rhythm, the activity rest rhythm. The rhythm that Ron Konopka and Seema Benzer used to screen flies that were mutated was the rhythm in emergence of adults from pupae. Here is a plot of emergence of adults from pupae across days. So there are 4 days, and these cultures are kept under constant darkness and constant temperature, so there's no information about time from the outside. These graphs may look a little different from the ones that Professor Ranganath showed. Here time on the x-axis is number of days, and you can see that emergence does not happen throughout the day and night, that there seems to be some gating, so that flies will emerge preferentially in a certain time window. Now if you look at these lines are drawn just for visualization, you can see that there are 4 peaks in emergence across 4 days. And now when they look at lines of fruit flies that had been mutagenized, they were able to identify 3 different variants. One variant looked like this, where there was no pattern in emergence. I hope it's evident that there is clearly no gating in emergence across days. There was another line where these flies finished 4 cycles in 3 days, and another line where in 3 days 3 cycles were not completed. And so they called these mutants as being arrhythmic, having a short period or having a long period. And remarkably, when they took individuals from each of these lines and measured their activity rest rhythm, so this is emergence, the adult coming out of pupae. When they took out flies from each of these and looked at their activity rest rhythms, here is a wild type. You can see a nice pattern, and that periodicity is approximately 24 hours under constant darkness. But if you took flies from this culture, they did not show a day-night pattern in their activity rest rhythm. Moreover, if you took flies which had a faster running emergence rhythm, it looked like these flies also had a faster running activity rest rhythm. Also, similarly, the ones with a longer running emergence rhythm also had a longer running activity rest rhythm. Eventually, they came to the conclusion, based on several other studies, complementation analysis, etc., that all of these mutations seem to have occurred at one locus on the X chromosome of the fly, and they were able to hypothesize that they have identified a gene which they termed the period gene, because a mutation in this gene seems to affect their ability to be rhythmic or not, and also the speed with which their clocks ran. So this happened in the year 1971 that this paper was published, but it was not clear at that point what this gene is doing in order to generate a clock. That work was initiated around the mid-1980s by the work by the studies of these 3 investigators, Jeff Hall, Michael Rosbash, and Michael Young, who shared the Nobel Prize this year. These 2 were friends and collaborators at Brandeis University, while Michael Young was at Rockefeller University, not that far away. But these 2 groups, when they began, were intense competitors, very bitter and acrimonious rivals to some point. However, possibly because of the intense competition, the speed with which research progressed and our understanding progressed may have been affected by the fact that they were competing very hard. However, I must add a cautionary note, and that now these 3 are all good friends. They also admit that perhaps they were a bit too rushed when they came to conclusions simply because they wanted to be the first to publish. And they came up to several erroneous conclusions along the way. Okay, so I'm going to very quickly tell you about what they found, starting with this one discovery that perhaps there is a location on the X chromosome of the fruit fly that determines periodicity and can have profound effects on whether a fly shows rhythmic locomotion, has a certain speed in that in the rhythm. So starting from simply the location of where this gene may be located, there were studies that both labs were engaged in, which is basically to sequence the gene. And they were both successful probably because they were in locations where the methodology for doing sequencing was highly developed. So where these results came from was highly influenced by the fact that that methodology was available in those places, and they were experts in the field, and they made use of it to address this question. So first, they looked at the sequence of the gene, which was not terribly useful, because from the predictions of what that protein was, they couldn't really tell why that protein should function as a clock gene. Next, they did some experiments where they put back genes. Again, this was possible with fruit flies because at that time, again, methodologies were available where you could do genetic manipulations and put back the normal gene into a mutant phenotype, and they were able to show that this gene indeed contributes to this phenotype. Next, they raised antibodies to ask where that protein is expressed and what is the nature of the expression of that protein. They were happy to note that indeed that protein is expressed in the brain, and moreover, the levels of that protein oscillates. So that's nice if you're looking at a clock which is showing, if you're trying to find a clock gene, and you're looking at rhythmic behavior, and you find that the levels in the protein also oscillates, it is heartening. Second, they were able to show that and I think one of the really major findings was that the messenger RNA levels also oscillates, and they were able to show that the gene or the sequence on the DNA of this gene has a role to play on determining the speed of its oscillation. Even more important, they were able to see that there was a difference in the phasing of the oscillation in the period protein and mRNA, and period protein oscillation was 6 hours delayed compared to the messenger RNA oscillation. They were able to also show that this protein shuttles between the cytoplasm and the nucleus, and then this suggested that there were suggestions that this protein may have some role to play in modifying transcription. So there might be some transcriptional regulational activity. Followed by the studies of period and also a little bit in parallel, other genes were discovered using similar methods, basically to do mutagenesis and look for variations in phenotype from normal to anything that's not rhythmic or having differences in speed of the clock. So there was timeless that was discovered by Amita Sehgal in Michael Young's lab, and there were several other genes that or mutations that were identified that affected clock phenotype. Also importantly, one photoreceptor was also identified by studies from these two groups, which showed that there was this one photopigment which was not important for vision but seems to be a dedicated protein for the clock to be able to see light. So I'm going to put the information that came out from all of these studies. Important to note that all of these discoveries made in fruit flies helped people who are working in parallel with mammals to be able to look for homologs and be able to piece together what the clock may be made of in mammalian systems as well. So I'm going to make a very simplified model of what they thought was happening with these genes and their proteins and their interactions being able to generate a self-sustained oscillation. Basically, that is what we want to get at. So here, this cartoon shows you on the left the nucleus of a cell, and on the right of this pink line is the cytoplasm. So the idea that they came up with is that the period gene, shown here, the coding region of the period gene is shown in white, and the regulatory regions upstream of the period gene are shown here with this E box. What they showed is that two proteins, clock and cycle, form a complex that comes and turns on the production of period mRNA. Now, period mRNA in the cytoplasm, and also the same is true for another gene called timeless, also is undergoing the same kind of regulation with clock and cycle. Now, there is spur and timeless mRNA in the cytoplasm that can become proteins, and these two proteins will have to heterodimerize, that enables these two proteins to remain stable with the help of several other post-translational modifiers such as DBT, such that this heterodimer enables the period and timeless complex to enter into the nucleus. Once inside the nucleus, this complex is capable of inhibiting its own transcription. In this manner, a loop is completed, which is negative because whenever there is period or timeless mRNA being produced, then gradually period and timeless protein can be produced. That protein, because of various lags that are built in, would require a certain amount of time for it to enter into the nucleus. Once inside the nucleus, if they are able to survive other degrading processes, can go inside and inhibit their own transcription, such that now period and timeless mRNA starts to drop. In this manner, a near 24-hour loop is completed, and this transcription translation feedback loop can give rise to a self-sustained oscillation. This is a very highly simplified version of how you can get a ticking clock. Now, this would need to be also synchronized by time cues from the outside, and light is a very important time cue. So, we will see what happens in the presence of light. We have the clock and cycle heterodimer turning on period and timeless transcription, which means you have period and timeless proteins. However, in the presence of light, this photoreceptor cryptochrome that I spoke about is capable of degrading timeless, such that there is not enough timeless protein in the cytoplasm, and hence the timeless and timeless and period proteins are unable to enter until the light goes off. So, that when there is no light, now timeless and period proteins can enter the nucleus, turn off transcription, and be able to complete the loop. In this manner, cycles of light and darkness can help to reset the clock. Now, that was, as I said, the highly simplified version. This is a slightly more complex version, but the real story is much gorier than that. But the point is that it is possible to build a self-sustained oscillation with the help of feedback loops, and it turns out that there are actually multiple feedback loops, and some of them are interlocked. I mean, they are interlocked because there are some components in these loops that are players in both loops, and such a system, a very similar system, was worked out to happen also in mammals. So, now what do we know is the genetic or molecular basis of circadian clocks? For the most part, the system is what I said just now. It is a system of transcription and translational feedback that is generating a self-sustained oscillation. However, there are exceptions to the rule. There are some systems in which, actually one species of cyanobacterium, in which a post-translational feedback is sufficient to generate a self-sustained oscillation. More recently, it has been shown that oxidation of another molecule, peroxyredoxin, can also be a player that can act as a self-sustained clock. And so, for the most part, we know that there are transcriptional translational feedback loops, plus there are cytosolic loops consisting of several small molecules, and they probably may even be more ancient than the transcriptional translational feedback loops. And the beauty, or the reason perhaps that this prize was awarded to these three researchers, was the fact that this particular system that can generate a self-sustained oscillation is found pretty commonly across a whole range of organisms. You can pick neurosporas, zebrafish, flies, cyanobacteria. A similar mechanism of transcription translational feedback loop, interlocked feedback loops, has been found to persist. Now, if one moves to complex organisms, where do these clocks lie? In humans, we know that the circadian, there is at least one important clock location in a particular region of the brain called the suprachiasmatic nucleus. The name is given based on its location. It is situated a little bit above the optic chiasm, and light that goes to the eye from the retina while going to the visual information processing areas also sends out certain nerve fibers that go into the suprachiasmatic nucleus and tells the clock that there is light outside. In this manner, the clock is reset. This master clock that sits in the suprachiasmatic nucleus sends out information to other body parts via nerves and also through hormonal substances. Moreover, in addition to this clock that sits in the suprachiasmatic nucleus, there are clocks that are known to exist in other tissues such as the liver, such as the kidney. These clocks don't necessarily listen to light as a time cue, but can use food, activity of the organism, and even body temperature as time cues. So the organization of clocks in living organisms now is not thought of as one clock, but more like a clock shop, where there are some clocks that are higher up in the hierarchy that sit in the brain. The suprachiasmatic nucleus is one, and there are several other regions in the brain especially of vertebrates, that can have self-sustained oscillators. In addition, there are peripheral clocks that are situated in several tissues. Depending upon which clock you're talking about, they may use information about time from different quarters. Okay, now we move to looking at what people are doing more nowadays, which is to move away from what people did in the past when they were trying to understand clocks, where they brought animals into the labs, studied them in somewhat sterile conditions and somewhat boring conditions, where you had one time cue or two time cues at the most, and asked how clocks respond to such time cues, to looking at clocks in more real world conditions. Here is an example of studies that have been conducted in Northern Europe, where they are trying to ask this fundamental question of why would you need to have clocks? Do clocks really give you an advantage? Do animals which have a good clock, are they at an advantage over animals that do not have clocks? And some emerging studies from here point out that perhaps clocks do give them an advantage, but the results are not really that dramatic to be able to say that clocks enable these, at least mouse species, to become better than their counterparts. Then there are people who put out running wheels in the wild to look at whether in the wild animals indeed show a rhythmic pattern in occupying these wheels, and whether there are different species which run these wheels at different times of the day, and so on. We also have an enclosure in our campus where we are looking at flies in somewhat more real world conditions, where they experience light, temperature, humidity, and other factors that cycle in nature. We are looking at behaviors at a very proximate level, and we are also trying to understand how fly populations that are out in such conditions change over time. That brings us to one other aspect, which is why should we study circadian rhythms? They are very fundamental, very fascinating phenomena that is good enough reason to study them. In addition to that, there is a more practical aspect of the fact that today's world, perhaps we don't listen to our clocks so much, and several occupations demand us to not be able to listen to the clocks that we've evolved with, and that leads to also several problems. And it helps to be able to study how aberrant behaviors can be slightly mitigated by knowing how our clocks work. There is a circadian prescription that many people give as to when one must do things at what time of the day and so on, which perhaps even our ancient Ayurvedic texts told us what to do, but our circadian biologists will tell you this as well. And some of you may disagree with some of the things that are shown here. However, you can be reassured by the fact that yes, there is actually a lot of variation in the human population. Most of us tend to wake up or like to wake up at a certain time of the day and go to bed at a certain time of the day. However, there are several individuals in human populations also that fall at the extreme and therefore probably suffer from occupational time keeping. One last point I would like to mention is that circadian clock gene mutations have been found to have associations with sleep phenotypes as well. And I will skip this quickly to show you that there are several circadian clock genes and mutations in them that are associated with sleep disorders. And those of you who are interested can look up the literature that's available online. So the take-home message I hope that you've been able to get from here is that clocks are endogenous. They are self-sustained oscillators that have temperature compensation. At the same time, they must be able to entrain to external time cues, and they are made up mostly of transcription translational feedback loops, and the organization is that of central and peripheral clocks, and that it's important to study clocks, and of course flies were very useful to study them. One last slide. I wanted to also show you that one of the Nobel Prize winners was here in this very hall a few years ago, and he was part of a course that was happening here. And I also wanted to remember two people of JNC who may have been very delighted by the fact that we are holding this symposium and by the Nobel Prize and the fact that you are all getting possibly infected by the research bug. This is the group at JNC who is interested in clocks. Thank you for your attention, and I'll be happy to answer any questions. Okay. Maybe
if there's a one quick burning question, maybe later then. Okay. Where? I can't see. Oh, give it to her.
My question is, we understood circadian rhythms, the molecular basis of it to be a kind of a transcriptional feedback loop that is 24 hours. The question here is like, in spite of having a day-light periodism, since it is fine-tuned to 20, should be observing this pattern irrespective of whether we expose it to light or night, and it should perpetually keep on maintaining itself. So why does it peter out when we remove these light cues?
It should not peter out when we remove time cues. Then only then we will be able to call it.
After a certain number of days, you showed that the data in the activity pattern kind of like petered out, kind of homogenized.
I'm not sure which one you're pointing to, but for a self-sustained clock, it should not peter out, otherwise it would be a damped oscillation. However, there would be limitation of a biological system, things like food availability and so on. Those things can lead to petering out of the system, lack of energy in the system. But otherwise, an oscillation in a clock must sustain for as long as the animal is alive.
Okay.
It could be simply wearing off of the machinery.
If we keep this nutrition and other things constant and just vary the light cue.
Vary the light cue, I didn't understand what you mean. Keep it constant, right?
Keep it constant, yes.
Give it no information about time from the outside.
Yes, exactly.
Yes, something will qualify as a clock only if that oscillation persists. Otherwise, it's not considered as a good clock.
Okay. Thank you.
Okay. Let us once again thank Professor Sheeba Vasu for this.